talk a little bit now about amino acid catabolism, how we can get energy out of our proteins. So proteins, first of all, are formed of carbon chains and an amine group, which I'm representing in this kind of dark maroon color. So the amine group contains nitrogen, and then we have these carbon chains. When we are using the word proteolysis, lysis meaning breaking apart, proteolysis is referring to taking a large protein and breaking it apart into its individual amino acid building blocks. And those amino acid building blocks, we can then take them apart and break them down for energy. So I'm going to walk you through how we do that here. This is just reiterating again that in my little demonstration amino acid, we have the amine group in maroon, and then we have a carbon skeleton in these teal balls. Now, the first step after we have gotten our individual amino acid is first we're going to take off that amine group. We're going to deaminate our amino acid, and then we're going to be left with just the carbon skeleton. Now, this ammonia, this amine group, is then going to be converted into urea. And this is really important because if we had a whole bunch of ammonia floating around, ammonia is toxic to the brain. So specifically, cells in the liver are going to take this ammonia after we've deaminated our amino acids, and it's going to convert it into a nice harmless compound called urea. And ultimately, we can excrete that urea in our urine. Okay, so now we've taken the amine group off of our amino acid. Now we're left with just the carbon skeleton. Now we know that there are 20 different amino acids, and they differ in the shape of their carbon skeleton. So amino acids come in 20 different flavors, 20 different types of carbon skeletons. Some of those carbon skeletons, they can be converted into pyruvate through some reshuffling, or they can be converted into intermediates that are part of the Krebs cycle. So carbon skeletons that can be either converted into pyruvate or into intermediates in the Krebs cycle, those are skeletons that ultimately can be used to build glucose through gluconeogenesis. Because remember, gluconeogenesis started with oxaloacetate, one of the Krebs cycle intermediates. So any carbon skeleton that can either turn into pyruvate or into the Krebs cycle intermediates, those are called glucogenic amino acids because they can be used to build glucose. However, there's another group of carbon skeletons that can't be converted to pyruvate or Krebs cycle intermediates, but they can be converted into acetyl-CoA. And remember, we know that in order to make ketones, we take two acetyl-CoAs and bind them and go through some chemical reactions to create ketones. So any amino acid carbon skeletons that can be converted into acetyl-CoA, those are called ketogenic amino acids. And so then from there, whether it's ketones or whether it's pyruvate, Krebs cycle intermediates, we can break down these carbon skeletons in order to synthesize ATP, going through the same types of redox reactions that we've talked about already. Now, typically, we are not going to rely on our amino acids as a source of energy. Usually, we are going to try and preserve our amino acids to be used to synthesize proteins, and instead, we will hopefully mostly rely on glucose and fatty acids for energy sources. So we're going to try and preserve our amino acids for protein synthesis.